particles settle to the bottom of the fermentation tank, and crystals of tartaric acid also sink to the bottom. The wine must be drawn out of the tank carefully without disturbing the sediment because wine with sediment is less appealing. The tartaric acid crystals are a hallmark of grape fermentation, and archaeologists search for them in old pots to ascertain whether particular peoples were producing wine at a certain time or place. Fermentation continues as long as sugar is present and the concentration of ethanol has not reached lethal levels. If all the sugar is fermented, the wine is a dry wine, but if some sugar remains, it is a sweet wine. Most wines produced in the United States have an alcohol content between 12% and 14% and have some sugar left, so their fermentation must be stopped artificially. This is usually done by microfiltering the wine to remove all yeast. The alternative is to heat the wine enough to kill the yeast, but that damages the flavor. At this point, the wine begins an aging process. White wines are aged only briefly, between 12 and 18 months, but red wines may be aged for up to 5 years. Typically, wine is aged in large oak barrels, where chemicals in the wine interact with compounds in the heartwood. These reactions alter the flavor and aroma of the wine and even create new types of longer-chain alcohols that give the wine a more viscous, substantial feel in the mouth. When the wine master has decided the wine is ready, it is transferred to bottles and either shipped to market or set aside for more aging in the bottles. Champagnes and sparkling wines are made by adding sugar to a bottle of wine that still has a few live yeast cells in it. The wine then continues to ferment, but all the carbon dioxide is trapped inside and builds up pressure. When the bottle is uncorked, the carbon dioxide forms bubbles and makes the wine effervesce. To be called champagne, the wine must be produced in the Champagne region of France. All other effervescent wines are sparkling wines, although those of Italy are usually referred to as Prosecco. Inexpensive sparkling wines are made by simply carbonating white wine in the same way that soft drinks are carbonated. If extra ethanol is added to a wine, it becomes a fortified wine. Examples are sherry, port, and Madeira. Such wines are then aged either in bottles with no exposure to oxygen or in oak barrels that permit a slight oxidation and evaporation, giving the wine a richer feel in the mouth. For example, tawny port is made by stopping fermentation while half the grape sugars are still present. Distilled spirits are added to bring its alcohol level to 20%, and then the wine is aged for 10 years in oak barrels that allow a very small amount of oxygen to act on the mix, converting it from wine to port. During this time, the bright red pigment of the grape skins precipitate and change color to a tawny brown. The effects of global climate change could be devastating for vineyards and wine production. Even with irrigation and a sunny climate, weather still plays a crucial role in the quality of the grapes. Vineyards are typically some of the most costly land used for agriculture, and all the production facilities are located close to most vineyards. Juice is rarely shipped more than a few miles. If an area's climate changes to one slightly warmer or wetter, if the date of the last frost of spring changes, or if rains fall at a different time, wine grapes could be rendered useless. Most existing vineyards have already been carefully chosen to be suitable for a particular variety of grape and for the production of a particular type of wine, so any change in weather or climate will almost certainly hurt the crop rather than improve it. Spirits. Spirits are alcoholic beverages with an ethanol content above 20%. Once fermentation has produced enough alcohol to raise its concentration to 20%, the solution is toxic to the yeast and they die. To obtain a higher alcohol content, either strong alcohol must be added or the solution must be distilled. During distillation, the solution is heated, which mostly causes the alcohol to evaporate, along with some water and some of the flavoring compounds. Distillation is possible because different liquids boil at different temperatures, and ethanol boils at 78 degrees Celsius, which is much cooler than the boiling point of water at 100 degrees Celsius. By keeping the mixture above 173 degrees Fahrenheit and below 212 degrees Fahrenheit, ethanol and certain flavors evaporate and most water remains behind. The vapors are carried away to a pipe that is chilled so the vapors condense into a new liquid, one that has a higher concentration of alcohol, less water, and an altered set of flavorings. This second liquid can be redistilled to raise the alcohol even higher and to achieve a different flavor. Distillation can raise the alcohol content only to 95.6%, making it 191 proof liquor. The proof number is twice the alcohol content. The type of spirit that results from distillation depends on the initial fermented material. Distillation of grain-based fermentations produces whiskies, vodka, and gin. For example, Scotch and Irish whiskies use primarily or only malted barley, but American whiskies use up to 80% corn as well, and rye and wheat may also be added. Gin also begins with the fermentation of malted barley, corn, and rye, but during distillation juniper berries are added for their flavor and aroma. Vodka is basically just pure ethanol and water. The objective of distillation is to raise the concentration of ethanol and to eliminate all flavors. Consequently, the initial fermentation for vodka is made with whatever carbohydrate is cheapest: wheat, corn, potatoes, and even sugar beets. Brandy results from distillation of grape wine. Distillation of fermented molasses creates rum, whereas tequila and mezcal are distilled from fermented juices of agave plants, tequila from agave tequilana and mezcal from A. americana. Both agaves are unusual in that they store their carbohydrates not as starch, as most plants do, but as inulin, a polymer of fructose. The agaves must be cooked to break the inulin down to simple sugars before fermentation can start. Tequila agaves are steamed, whereas those for mezcal are roasted slowly. Warnings. Fermentation of plant material always produces ethyl alcohol, and this is classified as a depressant. This seems counterintuitive, because light drinking causes people to be more lively and less inhibited, but even moderate drinking reduces a person's ability to focus their attention and slows their reaction speed. Drinking ethanol has both beneficial and harmful consequences. Moderate consumption, about 2 drinks per day, lowers the risk of heart disease. Red wine seems to be the most beneficial because it has resveratrol. In contrast, deaths related to ethanol, including driving while under the influence of alcohol, is the 5th greatest cause of death among North Americans. Also, drunk drivers don't just kill themselves, they kill and maim innocent bystanders. Ethanol is not at all addictive to many people, but it is to others and results in alcoholism. Ethanol easily passes from mother to fetus through the placenta and can cause fetal alcohol syndrome if the mother consumes too much ethanol while pregnant. The effect of ethanol on us depends on the amount we drink in proportion to our body size, which in turn determines how much ethanol passes into our blood. When the concentration of ethanol in our blood reaches 0.2%, it starts to interfere with both our coordination and our reaction time, and it may cause us to have impulsive behavior. At a BAC of 0.15%, people are drunk and have trouble walking or doing other simple things. A BAC of 0.4% is usually fatal. In the United States, it is illegal to drive if your BAC is 0.08% or higher. As a general rule, a person can metabolize the ethanol in one standard-sized drink in about 60 to 90 minutes. If you drink faster than that, chances are good that you will get drunk. Figure 11.25 Mezcal is made from agave americana, which has long, spiny leaves but which stores its carbohydrate in its broad, short stem. The leaves must be cut off, leaving just the stem, which you see here. These hearts are being transferred from the truck to a stone-lined pit. Burning charcoal will be added and the entire pit will be covered to allow the hearts to roast slowly, converting their carbohydrates to simple sugars that can be fermented. Ethanol is only one of many types of alcohol. Other types we encounter commonly are isopropyl alcohol, used as rubbing alcohol, and methyl alcohol, used as wood alcohol. All alcohols are poisonous, but our bodies are able to detoxify ethanol if we drink only small amounts. With larger amounts we develop a hangover, and even larger amounts cause death. 1 to several college students die every year from drinking so much ethanol that it disrupts the lipids in their cell membranes. Methyl alcohol is very toxic by itself, and our livers convert it into the even more poisonous substances formaldehyde and formic acid. At doses too low to kill us, methyl alcohol causes blindness. Isopropyl alcohol is converted to acetone in our bodies. You may be familiar with acetone as fingernail polish remover. At the next level. 1. Ecology of respiration. The enzymes involved in respiration respond to temperature. Respiration is more rapid when it is warm, slower when it is cool. Surprisingly, plants do not seem to control respiration very well, and on warm nights, they respire more than they need to, producing unnecessary amounts of ATP that are then wasted. In greenhouses and nurseries, temperatures are kept cool to prevent excess respiration, which is the same reason for refrigerating fruit and vegetables. With global warming, plants will respire more than they do now, and that may affect their fitness. This is a new concern and you may have trouble finding much information, but it will be worth your while. 2. Biology of mitochondria. Mitochondrial biology is more difficult to follow than plastid biology because mitochondria are almost invisible by light microscopy and never contain pigments or granules. But we know that mitochondria divide and multiply as cells divide and grow. In some algae, all mitochondria fuse into just one mitochondrion prior to cell division, then that mitochondrion divides into two when the cell does, and afterward each daughter mitochondrion breaks into numerous smaller mitochondria. There have been fewer projects to sequence the DNA of mitochondria in plants, but we are learning a great deal about the evolution, development, and function of mitochondria and their genomes. Our knowledge of their electron carriers and membranes is much greater than it was just a few years ago. 3. Will higher levels of carbon dioxide in the atmosphere inhibit respiration? At present, the concentration of carbon dioxide in the atmosphere is 400 ppm and is expected to reach 700 ppm by the end of this century. The rate of any chemical reaction is influenced by the concentration of the reactants and products, so the increased concentration of carbon dioxide may be expected to inhibit respiration. Experiments in laboratories show little effect, but field experiments, in which the atmospheric concentration of carbon dioxide is altered across a large experimental plot of land, show that plants do respond to elevated carbon dioxide. We are not certain what is happening, because increased carbon dioxide also alters cytosolic acidity, but this is an area you might want to investigate on your own. Summary. 1. Respiration provides a cell with both ADP and small carbon compounds that are important in various metabolic pathways. 2. The type of respiration carried out by a cell depends on environmental factors and the state of differentiation of the cell. 3. Anaerobic respiration, fermentation, is inefficient because only 2 ATP molecules are produced for each glucose respired. However, under anaerobic conditions, it is selectively more advantageous than its alternative, death. 4. Some plant and animal tissues are facultatively anaerobic, but no large plant or animal can live for prolonged periods without aerobic respiration. They are all obligate aerobes. 5. 3 common electron acceptors are oxygen, acetaldehyde, and pyruvate. Only oxygen is abundant and cheap and results in a harmless waste product, water. 6. During the complete aerobic respiration of glucose, electrons are removed and transported, ultimately, to oxygen. As oxygen accepts electrons, it picks up protons, and water is formed. 7. Aerobic respiration consists of glycolysis, the citric acid cycle, and oxidative phosphorylation via the mitochondrial electron transport chain. Passage of electrons through the electron transport chain creates a chemiosmotic gradient of protons and hydroxyl ions. 8. Some tissues of some plants generate heat by carrying electrons on an alternative set of electron carriers that do not pump protons. No ATP is formed, so all energy of NADH is released as heat. 9. The pentose phosphate pathway is a complex interaction of metabolites that can produce erythrose, ribose, or NADPH. 10. Respiration increases as temperature increases, within the range of approximately 5 degrees Celsius to 25 degrees Celsius. During warm days and nights, respiration is much more rapid than during cool periods. Important terms. Lactate obligate aerobes pentose phosphate pathway respiration. Respiratory quotient strict aerobes thermogenic respiration tricarboxylic acid cycle. Review questions. 1. Most plants store excess energy from photosynthesis. Name some times when recovery of stored energy may occur. Does energy recovery always occur in the same sites where photosynthetic capture occurred? 2. When a storage organ becomes active after dormancy and mobilizes its reserves, the starch is usually converted to sucrose for transport. Why is starch not transported? 3. The oxidation of NADH to NAD requires transferring electrons from it onto something else. What are characteristics the ideal recipient would have? What is the recipient actually used? What does it turn into as electrons are added to it? 4. Cellular respiration falls into two categories. What are the two categories? When is each one used? 5. Define obligate aerobe, obligate anaerobe, and facultative aerobe. Name some type of organism that is an example of each. 6. Under what conditions does plant tissue experience lack of oxygen? How is ATP generated from glucose without oxygen? 7. Glycolysis can proceed only if is available. If it cannot be regenerated from NADH, the organism will starve, despite having glucose available. 8. The reduction of NAD to NADH during glycolysis is a problem. Why is this less of a problem if roots absorb nitrates and sulfates or if the plant is synthesizing fatty acids? 9. Anaerobic and aerobic forms of respiration differ in the ultimate electron acceptors for each process. What is the electron acceptor in each? What are the advantages and disadvantages of each? 10. With oxygen present, not only can NAD be regenerated, allowing glycolysis to continue, but even more is formed in the regeneration process. 11. During anaerobic respiration in animals, the electron acceptor is pyruvate. What is the acid that is formed? During hard, fast exercise, your own muscles probably synthesize this acid. What is the sensation you feel? 12. Plants do not make the acid mentioned in question 11. What do plants make instead during anaerobic respiration? Pyruvate is converted to, and then NADH reacts with that, forming. 13. Considering the negative aspects of anaerobic respiration, how could natural selection have produced something so inefficient? 14. What are the three basic parts of aerobic respiration? Which steps occur in mitochondria and which in cytosol? 15. Are the initial steps of aerobic and anaerobic respiration similar or dissimilar? 16. For the citric acid cycle to occur, pyruvate must be transported from where glycolysis occurs, across the to the. It is transported as a 2-carbon fragment called acetyl. What is the name of the carrier molecule that transports it? 17. The text mentions that the benefit in the citric acid cycle is the generation of more ATP, yet at only one step is ADP produced. How is the citric acid cycle involved in more ATP production than just that? 18. Examine Figure 11, 13. Where are the electron carriers of the mitochondrial electron transport chain located, in the matrix, the membrane, or in the crystal lumen? As they carry electrons, some of them also deposit protons. Are the protons deposited in the matrix or in the crystal lumen? 19. What is a chemiosmotic potential in mitochondria? 20. The flow of protons from the crystal lumen to the mitochondrial matrix can be used to synthesize ATP. Describe how this occurs. 21. During glycolysis, is all of the energy of glucose converted to energy in ATP? Why do compost piles become warm? 22. Some plants generate large amounts of heat. Name 2 examples. How is the generation of heat beneficial to each of these plants? What is the name of heat-generating respiration? 23. The intermediates of all respiratory pathways can be used in to make various compounds. It is not that they will be completely oxidized to carbon dioxide and water. 24. The pentose phosphate pathway has some intermediates that are pentoses. What is a pentose? Name the one that is shunted into nucleic acid metabolism. 25. The 4-carbon sugar erythrose 4-phosphate is the starting material in the synthesis of many compounds. Name 2 important examples. 26. Consider a meristematic cell preparing for mitosis and a young xylem cell differentiating into a fiber or a tracheary element. Each uses the pentose phosphate pathway, but for different products. Explain why. 27. In most tissues, if the temperature is increased by 10 degrees Celsius, how much is the rate of respiration increased? 28. The total energy yield of respiration relates the number of ATP molecules synthesized per glucose molecule respired. What is the yield for anaerobic respiration? For aerobic respiration? For heat-generating respiration? 29. What is the definition of the respiratory quotient? The complete aerobic respiration of glucose consumes 6 molecules of oxygen and produces 6 of carbon dioxide. What is the RQ for this process? If you measured the RQ of a plant and found that RQ equals 0.7, would you suspect that the plant was respiring lipids or citric acid? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Chapter 12 Transport Processes. Chapter Opener Image. These leaves of camellia are sunburned because long-distance transport of water to them failed. During a prolonged drought, the soil became so dry that roots could not pull water from it and consequently could not supply the leaves with sufficient water. The guard cells of the leaves would have closed, helping to conserve water, but that is similar to when we are dehydrated and stop sweating. Evaporation can no longer keep organs cool. The leaf overheated and tissues died. Fortunately, the buds are well protected by bud scales, and if the drought ends in time, the buds will make new leaves. Outline. Concepts: diffusion, osmosis, and active transport. Water potential. Cells and water movement. Short-distance intercellular transport. Guard cells. Motor cells. Transfer cells. Long-distance transport, phloem. Long-distance transport, xylem. Properties of water. Water transport through xylem. Control of water transport by guard cells Box 12-1 Botany and Beyond, Water and Ecology Box 12-2 Plants and People, Farming Wastelands Box 12-3 Alternatives, Desert Plant Biology. Learning Objectives. After reading this chapter, students will be able to Define diffusion, osmosis, and active transport. Recall the 3 types of membranes. Explain the components of the water potential equation. State 3 important points regarding movement of water. Describe the changes in osmotic potential and pressure potential when a cell is placed in a water solution. Restate the importance of incipient plasmolysis. Summarize the relationship between guard cells, adjacent cells, potassium, and water potential. Explain the pressure flow hypothesis. Discuss the cohesion, tension hypothesis. Describe differences in water availability in various environments. Did you know? Air is usually so dry it can pull water out of plants, the same way it makes our skin dry and lips chapped. Water is pulled upward through plants, similar to lifting an entire icicle simply by pulling up on its top. In contrast, sugary syrup is pushed through phloem the same way that sweat is pushed through sweat glands. In late winter and early spring, maple trees conduct sugar through xylem rather than through phloem. Maple syrup is concentrated xylem sap. Concepts. One fundamental aspect of life itself is the ability to transport specific substances to particular sites, moving molecules against the direction in which they would diffuse if left alone. After death occurs, atoms, ions, and molecules diffuse, moving from regions of higher to lower concentration, and the organization of protoplasm decays, the disorder of the components increases. Diffusion also occurs during life but proceeds more slowly than the controlled and oriented transport processes that tend to increase the order within the plant or animal body. Transport processes consume energy, and many are driven by the exergonic breaking of ATP's high-energy phosphate bonding orbitals. Specific transport occurs at virtually every level of biological organization. Enzymes transport electrons, protons, and acetyl groups. Membranes transport material across themselves. Cells transport material into and out of themselves as well as circulate it within the protoplasm. Entire organisms transport water, carbohydrates, minerals, and other nutrients from one organ to another, between roots, leaves, flowers, and fruits. Plants have only a few basic types of transport processes, and the fundamental principles are easy to understand. They are grouped here into short-distance transport, which involves distances of a few cell diameters or less, and long-distance transport between cells that are not close neighbors. Many types of short-distance transport involve transfer of basic nutrients from cells with access to the nutrients to cells that need them but are not in direct contact with them. Such transport requirements arose when early organisms evolved such that they had interior cells that were not in contact with the environment. Short-distance transport became necessary to the survival of internal cells. Long-distance transport is not absolutely essential in the construction of a large plant. Many large algae have no long-distance transport, nor do sponges, corals, or similar animals. However, the ability to conduct over long distances is definitely adaptive, especially for land plants. Before xylem and phloem evolved, a plant's absorbing cells could not penetrate deep into soil because they would starve so far from photosynthetic cells, nor could they have transported their absorbed nutrients very far upward. Being limited to the uppermost millimeter or 2 of soil meant that absorptive cells could not reach more permanently moist, deep soil where there are more minerals. The uppermost layers dry quickly and free minerals are leached away by rain. With xylem and phloem, roots that penetrate deeply can be kept alive and their gathered nutrients can be carried up to the shoot. Vascular tissues make it selectively advantageous for shoots to grow upright, elevating leaves into the sunlight above competing plants. This elevation is feasible because photosynthetically produced sugars can be transported downward to other plant parts. Such elevation of photosynthetic tissues resulted in tall plants that could also place their reproductive tissues at a high elevation, enabling spores or pollen to be distributed widely by wind. After insect-mediated pollination evolved, it was adaptive to have flowers located high, in an easily visible position. The evolution of transport processes affected all aspects of plant biology and permitted later evolution of many new types of plant organization. Vascular tissues also act as a mechanism by which nutrients are channeled to specific sites, resulting in rapid growth and development of those sites. At certain times, nutrients are transported to apical meristems, promoting growth and leaf primordium initiation. At other times, nutrients are directed to flower buds or young fruits, and at other times, production of wood and bark is supported. The combination of short and long-distance transport has resulted in the ability of some plants to become large and complex enough to survive temporary adverse conditions, such as drought, heat, or attack by pathogens. Because almost everything transported by a plant is dissolved in water, the ability of water to move throughout a plant is important. Water is an unusual liquid, it is heavy and viscous, and it adheres to cell components as well as to soil, factors that affect transport processes. Related to transport processes are isolation mechanisms that inhibit movement of substances. Plants are adept at synthesizing organic polymers impermeable to a variety of substances. The epidermis with its cutin-lined walls keeps water in the shoot after it has been transported there by the xylem. The Casparian strips of the endodermis prevent diffusion of minerals from one part of a root to another. Isolation mechanisms are essential if transport is to be useful. Figure 12.1 This is an opuntia cactus with flattened stems and tiny leaves that will soon abscise. Xylem and phloem are arranged in a net-like pattern, with leaves located at every vertex of the net. Xylem is a means of long-distance transport, carrying water from the tips of this plant's long roots, up into and throughout the stem. Short-distance transport occurs as water moves out off xylem and then from cell to cell until it reaches the epidermis. This is the xylem of an opuntia cactus, as in. The soft parenchyma of its cortex and pith rotted away, then the epidermis fell off, leaving only the lignified vascular bundles. Diffusion, osmosis, and active transport. The first thing to consider is the mechanism by which material moves through a solution and crosses a membrane. The simplest method is diffusion, in which the random movement of particles in solution causes them to move from areas where they are in relatively high concentration to areas where they are in relatively low concentration. Diffusion through a membrane is technically known as osmosis. Membranes are of three types. Freely permeable membranes allow all solutes to diffuse through them and have little biological significance. Completely impermeable membranes do not allow anything to pass through and occur as isolation barriers. Differentially or selectively permeable membranes allow only certain substances to pass through. All lipid-protein cell membranes are differentially permeable. Hydrophobic molecules diffuse easily through any cell membrane, whereas many polar, hydrophilic molecules can cross differentially permeable membranes only if the membranes have special protein channels through which the molecules can diffuse. Water molecules, even though highly polar, pass through all membranes, but their movement is more rapid if the membrane has protein channels called aquaporins. Most membranes also have membrane-bound molecular pumps that use the energy of ATP to force molecules across the membrane. Even if that type of molecule is extremely concentrated on the receiving side, this is active transport. The molecular pump, which is a protein, binds to both the molecule and ATP when ATP splits into ADP and phosphate. The energy is transferred to the pump, forcing it to change shape, carry the molecule across the membrane, and release it. The membrane must otherwise be extremely impermeable to the molecule or it would leak back. Proton pumping in photosynthesis and respiration are examples of active transport. All cell membranes are important in transport processes. The plasma membrane governs movement of material into and out of the cell. Substances can move across the vacuolar membrane by either osmosis or active transport. The vacuole acts as an accumulation space for sugars, pigments, crystals, and many other compounds. The endoplasmic reticulum and dictyosome membranes transport material that then accumulates in vesicles. These vesicles may be relatively permanent, remaining in the cell for long periods of time, or they may be a means of intracellular transport, in which the vesicles migrate through the cytoplasm and fuse with another organelle. During fusion, the membranes merge and the vesicle contents are transferred into the organelle. This is a common means of moving material from the endoplasmic reticulum to dictyosomes or from either of these organelles to the cell exterior by fusion with the plasma membrane. During cell division, the new cell plate is formed by the coalescence of vesicles from both endoplasmic reticulum and dictyosomes. Figure 12. To imagine an angiosperm that has no vascular tissue. Leaves would produce large amounts of glucose, but because diffusion is slow over long distances, the sugar would diffuse out of the leaf only slowly and in small quantities. The shoot apex has almost no chlorophyll. If it had to depend solely on its own photosynthesis, growth, leaf initiation, and leaf expansion would be extremely slow. With vasculature, glucose can be transported from regions of excess to regions of need. Apical meristems and leaf primordia can thereby grow very rapidly. Vascular tissues also make the minerals gathered by an extensive root system available to regions that need them. Figure 12. 3. The cuticle, composed of cutin, is a waterproof epidermal layer that acts as an isolation mechanism, retaining water within the plant and keeping pathogens out. Figure 12. For these crystals are located in a cell vacuole. Calcium and oxalic acid were transported across the vacuole membrane by molecular pumps, concentrating them until they crystallized into these needle-shaped rhapide crystals. Water potential. Like any other chemical, water has free energy, a capacity to do work. For most chemicals, this energy is called its chemical potential. Because water is so important in botany, its chemical potential is usually referred to as water potential and has the symbol psi. Water potential, the free energy of water, can be increased several ways. Water can be heated, put under pressure, or elevated. The energy of water can be decreased by cooling it, reducing pressure on it, or lowering it. Water's capacity to do work can be changed in other ways as well. When water adheres to a substance, these water molecules form hydrogen bonds to the material and are not as free to diffuse as are other water molecules. Their capacity to do work has decreased. Consider a small beaker of water. If it is pure water, it can flow, move, dissolve material, and hydrate substances, but if a sponge is added, water molecules adhere to the sponge material and can no longer flow or easily dissolve things. If a large amount of sugar is added instead of a sponge, the results are the same. Syrup is just a sugar solution, but the water molecules in syrup have less capacity to do work than do the molecules of pure water. Water potential has 3 components. In this equation, psi is pressure potential, the effect that pressure has on water potential. If water is under pressure, the pressure potential increases and so does water potential. If pressure decreases, so do the pressure potential and water potential. Pressure can be positive or negative. Most liquids cannot be stretched very much, but because water is cohesive, it actually can resist considerable tension. When water is under tension, pressure potential is a negative number. Potential is measured in units of pressure, usually in megapascals or bars. 1 megapascal is approximately equal to 10 bars or 10 atmospheres of pressure. Pure water at 1 atmosphere of pressure is defined as having a water potential of 0. Psi is osmotic potential, the effect that solutes have on water potential. In pure water, no solutes are present and osmotic potential is given the value of 0. 0 MPa. Adding solutes can only decrease water's free energy because water molecules interact with solute molecules and cannot diffuse easily. Therefore, osmotic potential is always negative. If water molecules do not interact with the added molecules, the substance does not dissolve. It is important to be careful here. Adding acid to water only seems to make water more active. The solution may have more free energy than the water, but it does not have more free energy than both pure water and the original concentrated acid. Osmotic potential is related to the number of particles present in solution, that is, a solution composed of 2g of glucose in 100 milliliters of water has an osmotic potential twice as negative as a solution of only 1g of glucose per 100 milliliters of water. This has some unexpected results. If a molecule of starch containing 1000 glucose units is hydrolyzed to 1000 free glucose molecules, the osmotic potential of the solution becomes much more negative because there are now 999 more particles in solution than previously. Using terms such as increase, decrease, larger, and smaller can be confusing when dealing with negative numbers. If the osmotic potential goes from 0.01 to 0.1 MPa, is it increasing or decreasing? It is least confusing to use the terms more negative, less positive, and so on. Psi is metric potential, water's adhesion to non-dissolved structures such as cell walls, membranes, and soil particles. Adhesion can only decrease water's free energy, and thus, metric potential is always negative. In soils, metric potential is important because so much of the soil water is tightly bound to soil particles, but in living cells, metric potential usually is much less important than osmotic potential or pressure potential and usually is ignored entirely. The water potential equation for living cells is usually considered to be just psi equals psi. Table 12, 1 possible values of water potential components. Movement of water is related to water potential. Substances diffuse from regions where they are more concentrated to regions where they are more dilute. This can be stated more precisely for water. Water moves from regions where water potential is relatively positive to regions where water potential is relatively more negative. This statement contains several important points. 1. Water moves whenever there is a difference in water potential within the mass of water. All protoplasts are interconnected and most cell walls are fully hydrated, therefore, basically all of a plant body is one mass of water. Water can move between regions in the plant if the water potentials of the regions are not equal. As a consequence, the water potential of any particular cell may change many times a day as various parts of a plant lose or gain moisture. 2. If the water potentials of two regions are equal, the regions are in equilibrium, and there is no net movement of water. Water still diffuses back and forth, but on average equal numbers of water molecules diffuse into and out of a site. Unless frozen, water is always in motion, always moving within a plant from areas where it is abundant or under pressure to areas where it is rare or under tension. 3. Water potentials must always be considered in pairs or groups. Because water moves from one site to another, the water potentials of the two sites are important. Knowing one single water potential does not allow us to predict whether water will move. Temperature is not a factor because the solutions being compared are assumed to be at the same temperature. This is not strictly correct if the water potentials of leaves and roots are compared, but the difference is not significant. Figure 12, 5. The material on the left is potassium hydroxide, on the right is starch. They were photographed immediately after being exposed to air, while they were dry. Photographed after 1 hour in humid air. Water has moved from where it was more concentrated to less concentrated. The potassium hydroxide holds water by forming a solution with a very negative osmotic potential. Water is held to the starch by adhering to the long polysaccharide molecules. Water is not obvious in the starch, but think of saltine crackers left unwrapped. By adding salt to eggplant, water can be drawn from the tissues, making them easier to cook. Cells and water movement. Some examples may help. Imagine a cell with a water potential of 0.1 MPa. It contains solutes that cause the osmotic potential to be some unknown negative number. The cell is turgid and presses against the cell wall, but the cell wall presses back equally, causing pressure on the cell, and the pressure potential is some unknown positive number. We can ignore metric potential because it is usually such a small number. The osmotic potential, whatever it is, plus the pressure potential, whatever it is, equals 0. 1 MPa. Now imagine the cell being placed in a beaker of solution that also has a water potential of 0.1 MPa. The two water potentials are equal, the cell and solution are in equilibrium, and no net water movement occurs, the cell neither shrinks nor swells. Water molecules do move between the cell and the solution, but approximately equal numbers move in each direction every second. Figure 12.6 If a solution whose water potential is 0.1 MPa is connected to a solution whose water potential is 0.2 MPa, the two are not in equilibrium. Water moves from a region of relatively positive water potential to a region of more negative potential, as indicated. Water molecules move in both directions, but more move to the right in any particular instant. Both beakers must have solute dissolved in them, and the 0.2 MPa solution must have twice as much solute. Because the concentration of solute molecules in the left beaker is lower, they are less able to restrict the movement of water molecules than those in the right beaker. If the water potential of two solutions is identical, they are in equilibrium, and no net movement of water occurs. In each second, equal numbers of water molecules move to the right and left. A C of 1.0 MPa is very negative and has a strong tendency to absorb water, but we cannot be certain that water will move to it in this case. The right beaker may contain a solution with a C of 1.1 MPa. Now imagine the same cell placed in a solution with a water potential of 0.3 MPa. The water potentials are not equal, that of the solution being more negative than that of the cell, and thus, water moves from the cell into the solution. How much water moves? As water leaves the cell, the solutes that remain in the protoplasm become more concentrated. Because more solutes are present per unit water, osmotic potential becomes more negative. As water moves out, the protoplasm volume becomes smaller, so the protoplast presses against the wall with less force and the wall presses back less, therefore, pressure potential becomes less positive. Because both osmotic potential and pressure potential are decreasing, so is the water potential of the cell. At some point, the cell's water potential reaches 0.3 MPa and is in equilibrium with the solution, then net water movement ceases. Of course, as water moves from the cell into the experimental solution in the beaker, the solution becomes more dilute, and its osmotic potential and water potential become less negative. Its pressure potential does not change because pressure cannot build up in an open beaker. Therefore, equilibrium actually occurs slightly above 0.3 MPa, but because most beakers are much larger than most cells, the amount of water that moves is much more significant to the cell than to the beaker solution. Figure 12.7 If soil is watered well and then allowed to dry over a period of days, the water potential of the soil solution gradually and smoothly becomes more negative. Every day, leaves lose water more rapidly than xylem replaces it, therefore, leaves dry slightly, and leaf water potential becomes more negative. At night, stomata close and xylem transport rehydrates the leaf tissue, but in each cycle, leaves dry more than the previous night. This is not very serious until daytime leaf water potential becomes more negative than the wilting point. When leaves wilt, many metabolic processes are adversely affected. Wilting point varies from species to species and is much more negative for xerophytes than for mesic plants. After soil becomes extremely dry, even nighttime rehydration does not bring leaf water potential above the wilting point. The plant is at its permanent wilting point, and severe stress damage may occur. All growth stops, and leaves and developing flower buds or fruits may die and be abscised. Figure 12.8 A cell whose C is 0.1 MPa but whose values of psi or psi are unknown. If the cell is placed in a solution with a C of 0.1 MPa, the cell is in equilibrium with the solution and no net movement of water occurs. If the cell is placed in a solution with a C of 0.3 MPa, the cell loses water to the solution until the cell's water potential is also 0.3 MPa, even if the cell is killed by loss of water. The fact that the cell's water potential becomes more negative means that the osmotic potential or the pressure potential or both also become more negative. Figure 12:9. The molecules in this tabletop are exerting just enough pressure upward to counteract gravitational force. There is no movement upward or downward of the tabletop, and thus, forces must be in equilibrium. Adding several books increases gravitational force, but no net movement occurs. The tabletop is slightly bent, which stretches its molecules, but they resist just enough to counter the new force. Even more force is perfectly balanced by the tabletop. If the books were removed, the stretching on the table would stop, and the molecules would go back to exerting only enough pressure to counteract its own weight. The table would not fly upward. Consider the relative importance of osmotic potential and pressure potential in this example. In order for osmotic potential to become twice as negative, the cell has to lose half its water or double the number of solute particles. Either action is drastic. Almost any cell dies if it loses half its water, and thus, osmotic potential does not usually increase or decrease more than a few megapascals. Pressure potential can change enormously, however, usually with movements of only small amounts of water. Consider the top of a table. Its molecules are pressing upward with exactly the same force that gravity is pulling them downward. Placing a book on the table causes the table's molecules to exert more pressure upward as their bonding orbitals are stretched. Figure 12. The table changes the amount of upward pressure it can exert with very little change in shape. A similar process occurs in cell walls. Imagine placing the cell, now with a water potential of 0.3 MPa, in a beaker of pure water. Some water moves inward, diluting the solutes and causing the osmotic potential to become less negative, but the change is not significant. However, the extra volume of the water causes the protoplast to swell and press against the wall with more force. The wall presses back with equal force and pressure potential rises rapidly, even though only a small amount of water moves in. How high will it rise? We cannot predict its value. However, it will go high enough that osmotic potential plus pressure potential will equal 0, and the cell will be in equilibrium with pure water. Can a cell ever absorb so much water that it bursts? Animal cells often burst if placed in pure water, a process called lysis, but plant cells can never burst. Walls, either primary or secondary, are always strong enough to resist breakage by water absorption. Even the thinnest, most delicate walls of mature parenchyma cells can exert enough pressure on the protoplast to raise the pressure potential high enough to counterbalance osmotic potential, however negative it might be. Figure 12. 10. A healthy cell, turgid and full of water and protoplasm. It is swollen and firm, just like the cells of unwilted leaves. Its walls are stretched and are pressing back against the protoplasm that is pressing on them. If even a small amount of water enters the cell, the slight increase in volume causes the walls to stretch and push back, and pressure builds inside the cell. Consequently, psi becomes much more positive and so does the cell's water potential. Because of the slight increase in the amount of water in the cell, the salts, sugars, amino acids, and all other solutes are now slightly more dilute, therefore, psi becomes slightly less negative also, but this change is insignificant compared with the change in psi. Immature, growing cells have weak, deformable walls and cannot generate enough pressure to stop water absorption. The cell grows rather than bursts. Under these conditions, the cell may increase greatly in size. With such a large influx of water, solutes in the cell may become significantly diluted, osmotic potential and water potential may become less negative, and the cell reaches hydraulic equilibrium with surrounding cells and growth stops. In growing regions, however, such as the tips of stems and roots, and in expanding leaves, cells can keep their osmotic potential and water potential very negative despite the influx of water, either by actively pumping in solutes through the plasma membrane or by hydrolyzing giant starch molecules into thousands of glucose molecules. After the proper size is reached, growth can be stopped either by strengthening the wall so that it exerts more pressure and raises the pressure potential, or by stopping the import of solutes or the hydrolysis of starch, allowing the osmotic potential to rise. Either way, rising pressure potential or osmotic potential causes the cell's water potential to rise and reach equilibrium with the surrounding cells, stopping the net inflow of water. Botany and beyond. Box 12-1 Water and Ecology. Water is essential to life. The regions of the world that have no water have no life, whereas those areas that do have water, even just a little water, have at least a few organisms. Think about the various ways that water is available to us living creatures. Think about aspects of the world that we already know. Use our two analytical questions. What are the alternative ways in which water is present? What are the consequences of each alternative? The water available in water. Most of the world is covered with water. Oceans, lakes, marshes, ponds, rivers, snow fields, and glaciers occupy more of Earth's surface than does land. It might seem that there is plenty of water in water, but we know that water comes in a variety of forms: fresh, salty, brackish, and so on. Lakes and rivers contain fresh water, that is, water that has very few solutes dissolved in it. It starts out as rain, water that condenses from clouds and is basically distilled water, pure water. As it collects in streams, lakes, and rivers, this water continues to be more or less pure. Mountain streams are clear and clean because the water is too pure for algae to grow in it. Larger rivers are less pure because they have received runoff from fertilized fields, lawns, and golf courses, and cities dump their sewage into rivers. Sewage is always rich in dissolved minerals and organic chemicals, and river water often reaches a balance of water and minerals that supports abundant life. Algae grow so abundantly that they degrade the river. As the algae die, their bodies sink and are decomposed by bacteria, a process that uses up so much oxygen that fish may suffocate. This process is called eutrophication, but you are probably fortunate enough to have never seen a eutrophied river. In the 1960s, we realized that we could never keep all of our sewage and agricultural runoff out of rivers, but if we could at least keep phosphate levels low, we could prevent eutrophication. Algae cannot grow without phosphate. Most phosphate pollution was due to laundry detergents, and since the 1960s, we have switched to alternative detergents that are free of phosphates. Figure B12, 1A. The small part of Yosemite National Park has many microhabitats that differ in water availability. The waterfall has abundant water when the stream is flowing, but it goes dry or freezes at times. Its spray zone provides some water, but the cliff face is wet only during a rain. It has no moisture holding capacity. Trees grow where soil retains moisture longer than on the cliff face, but some areas with soil are in full sun, whereas others are in shade. Thus, rates of transpirational water loss differ. Oceans contain salt water. Rivers bring in water and dilute salts, but the water will evaporate, form clouds, and then fall as rain again and run to the oceans, carrying another load of dilute salts. This is a mechanism that moves salts from land to ocean and concentrates them. Currently, the salts in seawater are so concentrated that land plants and animals cannot use ocean water to hydrate themselves. We would die if we tried to survive by drinking seawater, just as plants would die if we irrigated them with it. Marine organisms have plasma membranes and vacuolar membranes able to move salts either into or out of the protoplasm as necessary to keep the cells properly hydrated. Seagulls have glands that purify seawater and throw away the salt, but we land organisms cannot do that. Figure B12, 1B. This cactus Armiticerius procerus grows in a remarkably dry area of coastal Peru. It is shriveled and sunburned from lack of water. It appears that this area receives rain only during El Niño years, and those occur only about once every 10 or 15 years. These plants must routinely go for 10 years or more with no rainfall whatsoever. Seawater's ratio of salt to water changes because of many factors. As rivers flow into an ocean, they deposit buoyant fresh water, which floats like a cap on the seawater. Marine algae cannot live in the cap of fresh water and must remain in the deeper saltwater, where they do not have as much light for photosynthesis. As the fresh river water spreads out, the thickness of its layer diminishes, and at some point, wave action mixes fresh and salt water together. The mouth of a river is a complex gradient of fresh and salt water. The complex mixing at the mouth of a river is a rich environment that provides numerous habitats for many types of creatures. Unfortunately, we have interfered greatly with these ecosystems. The simplest to understand are the Rio Grande as it flows into the Gulf of Mexico and the Colorado River as it flows into the Sea of Cortez. Both the United States and Mexico use so much of the river water for drinking, manufacturing, and irrigation that these two mighty rivers are nothing more than trickles that barely reach the coast. They no longer provide the millions of gallons of fresh water that would have maintained the coastal ecosystems, and without this outward flow of fresh water, the salty seawater approaches into the mouth of the rivers. Freshwater marshes have been destroyed by brackish water that floods in at high tide. The concentrations of salt and water in seawater also change greatly at the North and South Poles. The Arctic Ocean near the North Pole is frozen over with a sheet of ice, and at the South Pole, the continent of Antarctica is covered with snow fields and glaciers. Ice and snow are pure water, and as they melt in the summer, they dilute the salts in seawater. It may seem like these are such cold places that they must be lifeless except for a few polar bears in the north and penguins in the south, but that is not true. Polar water is so teeming with microscopic algae and animals that it is one of the most life-filled habitats in the entire world. The addition of fresh water as glaciers and icebergs melt in summer is a problem they have adapted to. In the winter, there is the opposite problem. As water freezes into ice, it leaves its salts behind. Therefore, as the poles become more ice-bound in winter, the remaining water becomes ever more salty, another dilemma for the creatures in it. An even worse problem is that the increasing saltiness causes increasing density, and the cold, salty water sinks. Fish and seals easily swim upward and remain safe, but algae and microscopic animals are carried downward into darkness where they become food for benthic creatures, those that live on the ocean floor. The intertidal zone, the region of coast that lies between the levels of low and high tides, is an area where available water changes greatly and quickly. As the tide goes out, sea anemones, barnacles, and algae are at first wet with seawater, but as the water on their surface evaporates, the concentration of salt increases. If exposed long enough, they may become not only dry but encrusted with salt, and they must retain water in their bodies against the osmotically dry salt on them. In contrast, a rain shower drenches them in fresh water, and they then struggle to prevent a loss of minerals out of their bodies. The water available in air. Air supplies water to land plants in the form of rain, fog, dew, frost, snow, hail, and even just humidity. Air also pulls water out of plants. It is the motive force for transpiration. Water availability is more complex than just the amount of rain that falls per year per square mile. Also important are the timing and regularity of precipitation, as well as the rate at which water is lost between rains. Some desert regions, for example, the Atacama Desert in Peru and Chile, are believed to have never received any rain at all, ever. At the other end of the scale are fog and cloud forests, regions where air is saturated most of the time, plants are dripping wet and soil is always moist. Far from being ideal habitats, fog forests are populated by dwarf trees, stunted because the humidity prevents transpiration and transport of minerals up from the roots. Most land habitats are more ordinary, having rainy days that alternate with sunny ones, but there are thousands of alternatives here: the ratio of rainy days to sunny ones, the amount of rain that falls each time, the seasons when it falls, the dryness of the air between rains, and so on. Who would guess that Seattle and Austin receive virtually identical amounts of rain? Each get about 35 inches of rain per year, but in Seattle, a bit falls every week, whereas Austin gets 0.6 to 6 inches every other month or so. Between rains, Seattle is cool and cloudy and plants transpire mildly, but in Austin, rains are separated by day after day of intense sunlight with low humidity, thus, plants transpire rapidly. Soils in the Pacific Northwest are thick and organic and hold rainwater in place for days, whereas Austin's soil is often less than 0.5 inch thick and it lies over porous limestone. Rainwater drains away immediately. Water is water, but there are many ways in which it is available to plants, animals, and other organisms. Likewise, there are even more ways in which organisms have become adapted such that they can use this essential resource. Figure B12, 1C cacti are not the only plants that survive drought by storing water in succulent bodies. The deserts of Africa and the Middle East have many succulent plants as well. This is Huernia recondita in the milkweed family, and there are also succulents related to geraniums and to dandelions among others. Figure 12, 11 Some aspects of biotechnology processes require that the cell wall be digested away with cellulase enzymes, leaving behind a naked protoplast. The digestion mixture must contain just enough solute, usually sucrose or the sugar alcohol mannitol, that the cells lose water and shrink away from the wall. If not, the protoplasts burst when the wall is removed. The solute concentration must be adjusted carefully. If too strong, too much water is pulled out of the cells and they die. Cells in suspension culture before treatment. Cells in 6% mannitol with the protoplasts just pulling back from the wall. Although plant cells cannot absorb so much water that they burst, water loss can be a serious problem. Imagine that our demonstration cell, now in pure water and with a water potential of 0.0 MPa, is placed in a strong sugar solution with a water potential of 2.0 MPa. Water moves out of the cell, osmotic potential becomes slightly more negative, and the pressure potential drops rapidly. In such a strong solution, long before the cell reaches equilibrium it loses so much water that the protoplast shrinks in volume and no longer presses against the wall. Plants never absorb so much water that their cells burst, but they frequently lose enough water to wilt because their protoplasts do not press firmly against the cell walls. The wall is not stretched and does not exert any pressure back, therefore, the pressure potential drops to 0.0 MPa. The point at which the protoplast has lost just enough water to pull slightly away from the wall is called incipient plasmolysis and is quite important. Up to that point, the cell has lost very little water, so its volume change and osmotic potential change have not been great, but because the pressure potential is now zero, the water potential equation is psi equals psi plus 0. If the cell has not reached equilibrium at the point of incipient plasmolysis, it continues to lose water, and the protoplast pulls completely away from the wall and shrinks. The cell has become plasmolyzed. Water potential continues to become more negative entirely because of the osmotic potential as solutes become more concentrated. Most plants at the equilibrium point of 2.0 MPa would die of severe water loss. Figure 12, when placed in a solution with a strongly negative water potential, the cell loses water rapidly and cell volume drops. Psi becomes less positive and cell becomes more negative. Psi changes only a small amount. Incipient plasmolysis is the point at which the protoplast has shrunk just enough to pull away from the wall, and thus, psi is 0 and cell equals psi. If the cell does not reach equilibrium at incipient plasmolysis, it continues to lose water, and cell continues to become more negative until it reaches -2.0 MPa. The pressure potential here cannot become a negative number, therefore, the changing water potential is due to a changing osmotic potential. During plasmolysis, the cell loses enough water to change the concentration of solutes significantly. Figure 13 These cultured cells have been placed in 12% mannitol for several hours and are severely plasmolyzed. After a few days of severe plasmolysis, the cells have died. Although such severe desiccation kills most cells, some can survive it easily. The embryos in most seeds are much drier, having water potentials as low as 20 MPa. Less dramatically, the leaves of desert shrubs in dry soil have water potentials as low as 2.0 to 6.0 MPa. For most plants of temperate climates, a leaf water potential below 1.0 MPa stops leaf growth, although leaves can survive such desiccation for many days or weeks. Table 12, 2 water potentials of various tissues under certain conditions. Short-distance intercellular transport. Most plant cells communicate with their neighboring cells, transferring water, sugars, minerals, and hormones at least. This movement occurs by a variety of mechanisms. First, all living cells are interconnected by plasmodesmata, the fine cytoplasmic channels that pass through primary cell walls. All of the protoplasm of one plant can be considered one continuous mass, referred to as the symplast. Material also is transferred from one cell to another by transport across the plasma membrane. The methods involve osmosis, molecular pumps in the plasma membrane itself, or fusion between transport vesicles and the plasma membrane. Once across a plasma membrane, a molecule initially resides in the cell wall. The wall is probably thin and permeable, and the molecule can penetrate it easily, diffusing across it to an intercellular space or laterally through it, spreading along the cell surface. Most small molecules can move easily through both the wall and the intercellular spaces. The two together are called the apoplast of the plant. In glands, the apoplast is mostly intercellular space through which molecules move easily, usually toward the surface of the gland. In non-glandular regions, the apoplast is mostly cell wall. The secreted molecule is probably absorbed by a cell neighboring the one that secreted it. In most parenchymatous tissues, primary walls are thin, and the contact faces between two cells are so extensive that the probability is much greater that a molecule will either diffuse more or less directly into the next cell or re-enter the cell from which it came. If the molecule was originally secreted by active transport, the original cell membrane is probably impermeable to it, at least in that area, so return to the original cell is usually not possible. This is probably the most common mechanism for movement of water, sugar, and other nutrients between parenchyma cells within cortex, pith, or leaf mesophyll. Guard cells. The opening and closing of stomatal pores are based on short-distance intercellular transport. At night, when stomata are closed, except for crassulation acid metabolism. Plants, guard cells are somewhat shrunken and have little internal pressure. They are in hydraulic equilibrium with surrounding cells, water enters and leaves guard cells at approximately the same rate, no net change occurs in the amount of water. When guard cells must open, such as just after sunrise, potassium ions are actively transported from surrounding cells into guard cells. Once inside the guard cells, the potassium cannot leave because the plasma membrane is impermeable to it. Potassium pumping is possible but diffusion is not. The loss of potassium causes the water potential in adjacent cells to become less negative, whereas absorption of potassium causes water potential in guard cells to become more negative. Adjacent cells and guard cells are thrown out of hydraulic equilibrium by potassium pumping, and water diffuses out of surrounding cells across their plasma membrane, across the two primary walls and middle lamella, and into guard cells across their plasma membrane. The extra water and potassium cause guard cells to swell, bend, and push apart, opening the stomatal pore. After they are open, potassium pumping stops. Water movement brings guard cells and adjacent cells into water potential equilibrium again, and net water movement stops. When guard cells must close, the process is reversed. Potassium is pumped from guard cells into surrounding cells. And water follows. Guard cells and adjacent cells are in equilibrium when stomata are fully open and fully closed. Figure 12:14. After being released from a cell, a molecule diffuses in a series of random short paths as it collides with and bounces off other molecules. Probability favors its entering a neighboring cell, but it can diffuse laterally along the wall as well. In many glands, the apoplast is large, so movement between cells may be faster and easier than movement within cells. Such glands often have a lining of compact tissue that isolates the gland, preventing the secreted material from permeating the entire region. This leaf tip of coastal sea oats is secreting water from a gland similar to that in Figure 12:15. At night, guard cells and adjacent cells are in hydraulic equilibrium, but at sunrise, potassium is pumped into guard cells, increasing solute concentration. Osmotic potential and water potential become more negative, and water flows in, causing guard cells to swell and open the pore. As pressure builds, pressure potential rises, counteracting the falling osmotic potential. Therefore, the guard cell's water potential rises and moves back into equilibrium with the adjacent cell, but by the time that happens, the stomatal pore is open. At night, when the stomata must close, all steps are reversed. Notice also that guard cells of fully opened and fully closed stomata are both in equilibrium with surrounding cells, even though they all have different internal conditions. When open, the guard cell's abundant potassium gives them a very negative osmotic potential, which is countered by turgor pressure, giving a large pressure potential. This results in only a small negative water potential. When closed, the guard cells have less potassium, therefore only a small negative osmotic potential. This is countered by less turgor and therefore a less positive pressure potential. These cells too have only a small negative water potential. Even though two cells might have very different internal conditions, they can have the same water potential and be in hydraulic equilibrium. Plants and people. Box 12-2. Farming wastelands. It has been proposed that we develop plants that tolerate high levels of salt and use them as crop plants in arid, marginal wastelands where fresh water is scarce. This might be possible in two ways. We could examine desert-adapted plants to see whether any have useful properties such as nutritious seeds, medicinal drugs, or useful fibers. Alternatively, desert-adapted plants could be studied to identify which of their features make them drought-resistant, then the corresponding genes could be identified, cloned, and transferred to some of our crops that are not now drought-adapted. Once either of these types of plants is available, they could be grown in semi-desert regions or perhaps even in deserts using a little irrigation with seawater or brackish water. This concept has many problems. The first is that it is one thing to find plants with enough drought and salt tolerance to grow in seawater during a 1-year-long experiment, but it is another thing to irrigate an area with seawater year after year. Because they have little rain, most arid regions have only small, temporary rivers that have not been able to carve effective drainage channels. They often end in dry lakes with no outlet. After a rain, water is present for a few weeks, but it evaporates, leaving its minerals behind. The rivers do not carry minerals out to sea continuously as do rivers in the eastern United States and other moist areas. If seawater is used for irrigation in an arid area, tremendous amounts of salt would be deposited as the water evaporates. This would continue year after year until the salt concentration becomes so high that a salt desert is created, such as occurs in the Great Salt Lake in Utah, the Dead Sea in the Middle East, or the Devil's Golf Course in Death Valley, where accumulated salt is so abundant it forms crystals 2 or 3 feet tall. Nothing could grow, not the new crop plants, not the genetically engineered plant, and certainly not the original plants, which would all be destroyed when the region was plowed to start the project. Even using fresh water rather than seawater causes a gradual accumulation of salt, and California's Central Valley is already facing a serious problem of salt accumulation in the soil. Another problem with this type of project is the concept that deserts and semi-deserts are wasteland, that they must be developed and improved. Although this type of thinking may have been popular at one time, many people now disagree with it. These areas, in their natural state, have an intrinsic worth. They are home to a great diversity of plants, animals, fungi, and other species. Even if none of these wasteland species is ever discovered to contain a medicinal drug or other useful feature, does that give us the right to exterminate them? Is it really necessary to bring more land into cultivation when so many Americans are overweight or eat foods that have been processed specifically to remove calories from them? Figure B12. This desert would be considered wasteland by some people, but it is where many rare and unusual plants live. This area has not only high biodiversity but also peace, quiet, beauty, and a chance for people to reconnect with nature. The plants that look like upside-down green octopuses are ocotillos. Fortunately, this wasteland is protected from developers. It is part of Big Bend National Park. Figure 12:16 Each leaflet of the compound leaf of oxalis is joined to the petiole by motor cells. Here they were photographed in the morning when it was cool, and the sunlight was not intense. Motor cells are turgid and leaflets are held into the sunlight. Oxalis plants in full, intense sunlight. The motor cells have lost potassium and water, thus, they are not turgid. Leaflets hang down, minimizing their exposure to light. Later in the afternoon, when sunlight is not so intense, or if the shadow of a tree moves across the plant, the motor cells will absorb potassium, then water, and raise the leaflets again. Motor cells. The leaves of sensitive plant, prayer plant, and many other species move slowly and reorient themselves by flexing and folding in response to a variety of stimuli. The location of flexure is either the entire midrib or the point at which the petiole attaches to the lamina or stem. The cells at these joints, called motor cells, are similar to guard cells. They can either accumulate or expel potassium and thus adjust their water potential and turgidity. In Venus flytrap, the leaf can close rapidly, in less than a second, but it requires several hours to reopen. Motor cells are located along the midrib, and when they are shrunken, pressure in other midrib cells causes the two halves of the blade to be oppressed, and the trap is closed. Trap opening occurs as potassium is slowly accumulated by motor cells, water diffuses in, and the motor cells become turgid. Closure is not caused by pumping potassium out of motor cells, that would be too slow. Instead, the membrane suddenly becomes freely permeable to potassium, and it rushes out instantly. The water balance is rapidly changed, and water too floods out, allowing the motor cells to virtually collapse. The trap then shuts quickly enough to catch insects. Transfer cells. The rate at which material can be actively transported depends on the number of molecular pumps present, which in turn depends on the surface area of the plasma membrane. The larger the membrane, the more molecular pumps it can hold. In certain specialized transfer cells, the walls are smooth on the outer surface but have numerous finger-like and ridge-like outgrowths on the inner surface. The plasma membrane is pressed firmly against all of the convolutions and thus has a much larger surface area than it would if the wall were flat. Consequently, room is available for many molecular pumps, and high-volume transport can occur across these transfer walls. Transfer cells are found in areas where rapid short-distance transport is expected to occur, in glands that secrete salt, in areas that pass nutrients to embryos, and in regions where sugar is loaded into or out of phloem. Long-distance transport, phloem. Although the exact mechanism by which water and nutrients are moved through phloem is not known, most evidence supports the pressure flow hypothesis. Membrane-bound molecular pumps and active transport are postulated to be the important driving forces. The sites from which water and nutrients are transported are sources. During spring and summer, leaves are dominant sources, as their photosynthetically produced sugars are exported to the rest of the plant. At other times, such as early spring, before new leaves have been produced by deciduous trees, the sources are storage sites such as tubers, corms, wood and bark parenchyma, and fleshy taproots. Cotyledons and endosperm are sources for embryos during germination. Within sources of many species, sugars are actively transported into sieve elements, sieve tube members in angiosperms, sieve cells in plants other than angiosperms. In other species, phloem is loaded by the polymer trap mechanism. Conducting cell plasma membranes are permeable to monosaccharides and disaccharides but not to polysaccharides. Simple sugars merely diffuse into the conducting cells and then are polymerized into polysaccharides that cannot diffuse back out. In both loading mechanisms, cells surrounding sieve elements, both companion cells and other phloem parenchyma cells, are important in loading phloem. Many of the cells are transfer cells. It is common now to think of the functional unit as consisting of both a conducting cell and one or several companion cells, and the term STMCC complex is used. As sugars accumulate in sieve elements, the sieve element protoplasm becomes more concentrated. Consequently, both its osmotic potential and its water potential become more negative. This causes hydraulic disequilibrium between sieve elements and surrounding cells, and water diffuses into the sieve elements. In any other cell, the increased volume of sugars and water would cause the protoplast to expand and press against the cell wall, but sieve elements are unique, being living cells with relatively large holes in their walls, up to 14 mm wide in cucumbers and pumpkins. When pressure starts to build in these cells, protoplasm is squeezed through the sieve pores into the next cell. Sieve element protoplasm is not like that of most cells, the vacuolar membrane disintegrates, allowing vacuolar water to mix with part of the cytoplasm, creating an extremely watery, non-viscous substance, phloem sap. The majority of the protoplasm is held firmly to the walls, probably by microtubules or microfilaments, and is not carried away with the watery central phloem sap. In sources, phloem loading occurs along numerous vascular bundles such as the fine veins and leaves, the network of bundles in tubers and corms, and the inner bark in storage roots and stems. With this massive loading, pressure builds quickly, and a large volume of material flows from the source. Pressures as high as 2.4 MPa have been measured in some sieve tubes. The rate of transport can be high, up to 660 cm HR has been measured in leaves of corn. The actual amount of sugars and other nutrients transported by phloem per hour is called the mass transfer. Vascular bundles vary not only in the speed at which their phloem translocates but also in the amount of phloem present. The number of bundles leaving a source is also important. To make comparisons easier, mass transfer can be divided by the cross-sectional area of phloem to obtain the specific mass transfer. Sinks are sites that receive transported phloem sap, and they are extremely diverse. Storage organs are important in perennial plants during summer, but also important are meristems, root tips, leaf primordia, growing flowers, fruits, and seeds. On even a small tree there may be thousands of sinks, each receiving nutrients. Not all sinks are active simultaneously. Most plants do not produce flowers and leaves at the same time, and fruits can develop only after flowers. Within sinks, sugars are actively unloaded from sieve elements into surrounding cells. The loss of sugar causes phloem sap to be more dilute, and its osmotic potential and water potential tend to become less negative, thus, water diffuses outward into the surrounding cells. As a result, even though phloem sap flows rapidly into a sink, the end cells of the phloem do not swell. As quickly as nutrients are loaded at sources, they are unloaded at sinks. Figure 12:17 As sucrose is actively transported into sieve elements, Cy and cell become more negative, moving away from equilibrium with companion cells and other neighboring cells. Water moves into the sieve elements, squeezing phloem sap out through the sieve pores. Because the sugary water escapes through sieve pores, pressure does not build high enough to stop the influx of water. Water potentials of sieve elements and surrounding cells never reach equilibrium as long as sucrose is being pumped. In sinks, sucrose is actively transported out of sieve elements, and all processes work in reverse compared with sources. Table 12, 3 Speed of phloem sap translocation. Figure 12:18. When this material of squash was being prepared for microscopy, it was cut open, causing the phloem sap to surge toward the cut, sweeping pea protein along and forming pea protein plugs, visible as dark brown masses. Sieve pores in the sieve plates are also visible. Table 12, for specific mass transfer. Because phloem sap is under pressure, the danger exists of uncontrolled bleeding if phloem is cut. Vascular bundles are broken open frequently, especially by chewing insects and larger animals. Two mechanisms seal broken sieve elements. The first is pea protein, found as a fine network adjacent to the plasma membrane inner surface of uninjured sieve elements. When phloem is ruptured, the phloem sap initially surges toward the break. This rapid movement sweeps pea protein into the cell center, where it becomes a tangled mass. When it is carried to a sieve area or sieve plate, the pea protein mass is too large to pass through and forms a pea protein plug. Pea protein is present in all eudicots and many but not all monocots. Conifers do not produce pea protein. Within uninjured phloem there is another polymer as well, collose. Apparently, it stays in solution only if it is under pressure. When injury causes a pressure drop, collose precipitates into a flocculent mass and is carried along with the pea protein to the nearest sieve areas. There the collose contributes to the plug, and leaking is prevented. In monocots with long-lived stems, such as palms and Joshua trees, sieve tube members live and function for many years, even hundreds of years, but in all other plants, individual sieve elements have a lifetime of only months or even weeks. They stop transporting and are replaced by new phloem cells from the provascular tissues or vascular cambium. After they cease to function, collose deposits seal them permanently. A further aspect of phloem transport is important to consider. As sugar is actively transported into phloem in sources, what happens to the water potential of the cells losing the sugar? Shouldn't it become less negative? No, it remains unchanged, because the sugars are being exported at the same rate they are being synthesized in leaves. Chlorenchyma cells absorb carbon dioxide molecules, but this does not cause the water potential to become more negative because the carbon dioxide is synthesized into sugar and exported. Millions of molecules of carbon dioxide may pass through a chlorenchyma cell without any long-term impact on its osmotic or water potentials. In sources such as tubers, sugar export has no impact on the storage cell water potential because as rapidly as it is exported, new sugar appears by depolymerization of starch. The same is true at sinks. Storage cells do not accumulate sugar as sucrose but instead polymerize it into starch. Thousands of molecules may be absorbed, but because they are polymerized into one molecule, no change in water potential occurs. In growing cells of sinks such as meristems and buds, the imported sugar is polymerized to cellulose, hemicellulose, and other carbohydrates. It can also be metabolized into amino acids, fatty acids, and nucleotides. These are then polymerized into proteins, fats, and nucleic acids, and thus, again, little or no change in osmotic potential and water potential occurs. In all cells, part of the imported sugar is respired, but this converts it to carbon dioxide that is expelled, so again there is no osmotic effect. Plants control the direction and rate of flow of phloem sap. While dormant in late winter and early spring, buds receive very little phloem sap, but after they become active, phloem transport increases greatly. Not all buds are equally affected. Some grow rapidly whereas others, even though located quite close by, receive virtually nothing. While flowers are open, phloem transport is low, but after fertilization, when the ovary begins to develop into a fruit, transport increases. The direction of transport can also change. Leaf primordia and young expanding leaves are sinks. Imported sugar allows them to develop much more rapidly than they would if they had to be completely autotrophic. In addition, they also need large amounts of nitrogen, sulfur, potassium, and other minerals. After leaves reach a critical size, they become self-supporting, able to photosynthesize rapidly enough to meet all their own needs. Shortly afterward they become sources, exporting material. Phloem transport is reversed in the leaf and petiole. Molecular pumps must now load the phloem, not unload it. Plasma membranes may be altered, or one set of sieve elements may cease to function and may be replaced by an entire new set of cells. The early primary phloem often lives for less than a few weeks. Figure 12:19. This bean contains two prominent sources, the cotyledons, which are supplying sugars, amino acids, and minerals to the rest of the seedling. The shoot tip with its meristem and leaf primordia are sinks, as are the roots. The first two leaves are expanded and are probably sources now, but they were sinks while they were developing. Leaves become self-sufficient and stop importing sugars when they have grown to about 1/4 of their mature size. At this point, the minor veins have not yet formed, so phloem unloading must occur by higher-order veins, not by the minor veins. As leaves continue to mature, minor veins are finally established, and the phloem in them is then used for export of material out of the leaf. Leaves near a stem tip export upward to the shoot apical meristem, while leaves farther back export toward the trunk and roots. As the shoot apex grows, leaves that had been near the apex are left behind and their transport shifts from upward to downward. If the apex produces flowers and then fruit, the direction of transport may shift again so that all leaves send sugars upward. Long-distance transport, xylem. Properties of water. The movement of water through xylem is based on a few simple properties of water and solutions. One property is that water molecules interact strongly with other water molecules, behaving as if weakly bound together. When frozen, the molecules become strongly bound to each other. Because of this, liquid water is said to be cohesive, and any force acting on one molecule acts on all neighboring ones as well. Another property of water is that its molecules interact with many other substances. It is adhesive. Almost all substances in plants, except lipids, interact with water. Cellulose, enzymes, DNA, sugars, and so forth have a shell of water molecules rather firmly attached to them. Occasionally, a water molecule vibrates out of one of these shells and is replaced by another water molecule, but in general, adhesion makes these water molecules less free to move around than other water molecules. Water also adheres firmly to soil particles. When soil is quite moist, roots can absorb the free liquid water between soil particles, but as the soil dries, the remaining water adheres firmly to the soil and cannot be absorbed easily, if at all. Even though the soil may contain considerable water, it is unavailable to the plant. The same is true of seawater. Water molecules interact so strongly with the salt molecules that land plants cannot pull the water away. Another property of water is that it is heavy, and lifting it to the top of a tree requires a great deal of energy. If water were lighter, less energy would be involved. Water transport through xylem. Water movement through xylem and plants as a whole is governed by the principles of water relations just described. The cohesion-tension hypothesis is the most widely accepted model of the process. When stomatal pores are open, they unavoidably allow water loss. The apoplastic space of spongy mesophyll and palisade parenchyma is filled with moisture-saturated air, so water molecules have a strong tendency to diffuse from intercellular spaces to the atmosphere. Even relatively humid air has a tremendous capacity to absorb water. At 50% relative humidity, warm air can have a water potential as negative as -50. 0 MPa. This water loss is called transstomatal transpiration. The cuticle and waxes on the epidermal surfaces are fairly efficient isolation mechanisms, being so hydrophobic that very little water passes through them. However, some water is lost directly through the cuticle by transcuticular transpiration. Figure 12:20. Wet soil contains water both as a film covering all surfaces of soil particles and as small masses held in capillary spaces formed where soil particles touch. The latter is held weakly and can be easily absorbed by roots. Dry soil contains only tightly bound films of water. This adhesion is measured by psi, which can be so negative that the soil's water potential is also very negative. This water cannot be absorbed by the roots of most plants. Consider a leaf in early morning, stomata are closed, air is cool, and relative humidity is high. The air may have cooled enough during the night to allow dew to form. Cells within the leaf are turgid and probably in equilibrium with each other, all having a water potential between 0.0 and 1.0 MPa. As the sun rises, stomata open and begin losing water, the air warms, and its relative humidity decreases. As transpiration causes epidermal cells and mesophyll cells near stomata to lose water, their water potentials become more negative, going out of equilibrium with surrounding cells. The disequilibrium does not become major because water diffuses into these cells from other cells and apoplastic spaces deeper within the leaf. But this water movement out of the deeper mesophyll cells causes their water potentials to become more negative, away from equilibrium with even deeper cells. Table 12, 5 Transstomatal and transcuticular transpiration. Rates are mgh/dm hour. Surface area includes both sides of the leaf. Finally, this gradient of water potentials reaches a tracheid or vessel member. As water molecules move out of tracheary elements into mesophyll parenchyma cells, the water potential within the xylem water column becomes more negative. The loss of water from tracheary elements does not really affect the xylem osmotic potential because solutes are very dilute to begin with. Here water's cohesive properties are more important. As a water molecule leaves the xylem, it does not leave a hole behind but instead drags other water molecules along with it. All water molecules of the plant are hydrogen bonded together, but the water molecules in the xylem can move upward most easily. That water is purest, is not bound to proteins and cellulose, is not locked into hydration shells around solutes, and so on. As water molecules diffuse out of xylem in the leaves, cohesive forces pull water upward through the xylem, all the way from the roots. Think of an icicle. If the top molecules are pulled upward, the entire mass of icicle is lifted. Water is heavy, and water molecules in the uppermost tracheary elements must lift the weight of the entire water column. There is tension on these molecules, and consequently, the pressure potential is a negative number. As water moves into the leaf mesophyll, the xylem water potential becomes more negative because of an increasingly negative pressure potential. In vertical stems, water must move directly upward in the xylem, and the water's weight is a significant consideration. In the examples discussed earlier, water could move laterally between a cell and a beaker, therefore, no lifting was involved. In vertical xylem, the weight of water counteracts its tendency to rise into areas of more negative water potential. Consequently, if leaf xylem water potential is only slightly more negative than root xylem water potential, the water does not move. For every 10 meters of height, leaf water potential must be at least 0.1 MPa more negative than root water potential. In trees such as elms and sycamores that are typically more than 30 meters tall, leaf water potential must be at least 0.3 MPa more negative than root water potential simply to overcome the weight of water. This is accomplished automatically. When stomata open in the morning, leaf cells lose water, and their water potentials become more negative. However, water does not begin moving upward in the xylem until drying causes the water potential of leaf cells to become sufficiently more negative. Stolons, rhizomes, and horizontal vines have no such problem. Long-distance transport is horizontal and no lifting is involved, so gravity is not a factor. A few plants grow as pendant epiphytes, their stems dangling down from the branches of the host plant. Their stems and leaves are lower than their roots, and gravity assists water movement. Water is extremely adhesive, and its molecules interact strongly with the polymers of the cell walls of tracheids and vessel elements. Water molecules adjacent to the walls tend to remain fixed to the walls and also tend to prevent neighboring water molecules from being drawn upward by transpiration cohesion. This results in a layer of relatively immobile water that does not move easily. In narrow tracheids and vessel elements, this immobile water is a significant fraction of the water column. The resulting friction hinders water's movement and contributes to the tendency of water to remain stationary even when leaves have a more negative water potential than roots. Imagine lifting an icicle. You must pull against its weight and the friction of the icicle in a tube, but lifting the top of the icicle raises the entire water column unless it breaks. As a rough approximation, to overcome friction, leaf water potential must be at least 0.1 MPa more negative than root water potential for every 10 meters of height. Therefore, considering both friction and gravity, a difference of 0.2 MPa is needed for every 10 meters of height. In plants with numerous wide vessels, friction is less, and less than 0.2 MPa is needed. However, in plants with narrow tracheary elements, even more than 0.2 MPa is necessary. Also, in plants that have only tracheids, water molecules must be pulled through pit membranes when entering and leaving each tracheid, further contributing to friction. Alternatives. Box 12-3 Desert Plant Biology. Plants in many habitats have more than enough water, their leaves do not wilt, and they do not abort flowers or fruits because of insufficient water. Plants adapted to deserts, however, must survive periods when moisture is so scarce that more water is lost from the plant than is taken in. What alternatives make life possible in dry regions, and what are the consequences of each alternative? What do we mean by desert or dry environment? Dryness results from the balance between precipitation, evaporation, and soil texture. Precipitation may fall evenly throughout the year or seasonally, often as winter snow or summer thunder showers. In some habitats, rain occurs as drizzle lasting for days or weeks and is accompanied by cloudy, cool weather. In others, it comes torrentially, with 2 to 8 inches falling in a single day, followed by bright sunny weather. Periods without precipitation might last just a few weeks, but it is not unusual for rainless periods to last months in the Chihuahua, Sonora, and Mojave deserts of the southwestern United States. Evaporation is related to the relative humidity of air. Hot air can hold enormous amounts of water vapor, cool air can hold less. Precipitation occurs when air cools so much that its holding capacity drops below its actual moisture content, excess water vapor. Condenses as fog, dew, rain, or snow. However, even cold air can be dry, and hot air can be humid. Plants face water problems when periods of low precipitation coincide with periods of dry air. Texture affects a soil's capacity to store precipitation. Rocky, sandy soils, especially those on slopes, hold almost no water and become dry just days after a heavy rain. Fresh lava flows in Hawaii are deserts despite receiving rain almost every day. Alternatively, fine soils with some clay and abundant humus hold large amounts of water for months. Roots have a steady supply to draw from, but even this is a subtle complexity for plants. Sandy desert soils are actually beneficial because they allow even a light rainfall to penetrate down to the root zone. If the desert had rich soil with clay and humus, light rains would be held in the uppermost layers, leaving roots dry. Averages are not especially helpful in understanding deserts and desert-adapted plants. Variations from year to year are important. The vegetation of any desert is the result of plants surviving not merely the droughts of average years but also rare protracted droughts that last several years. For example, cacti dominate the deserts of Mexico and the American Southwest because they survive exceptionally long droughts that kill off any plant that can only survive average droughts. If every year were an average year, these plants would become so abundant that they would overgrow the cacti, shading them and ultimately killing them. Two alternatives by which plants adapt to dry habitats are drought avoidance and drought tolerance. Most deserts have either a brief period when they are moist or they have small areas where water collects. Many drought-avoiding plants are known as desert ephemerals. They are small plants that complete their life cycle in just a month or two. Seeds germinate, seedlings grow, the plant flowers, produces new fruits and seeds in just a few weeks, while the soil is moist after a rain or a snow melt. By the time drought arrives, the plants have died, but their seeds are ready for the next moist season. These plants avoid dry conditions. Two consequences are that these plants can live in deserts and can never become large or perennial. Drought-tolerating plants live through dry periods, losing water more rapidly than they gain it. Most accomplish this by being succulent. A high percentage of their body consists of water storage cells. Stem succulents such as cacti, euphorbias, and stapeliads store water in pith and cortex. Leaf succulents such as agaves, yuccas, echeverias, and lit hops have very thick leaves. Desert-adapted bulbs are leaf succulents in which the upper portion of each leaf extends above ground and is thin and photosynthetic, whereas the lower portion of each leaf is subterranean, thick, fleshy, and able to store water and nutrients through adverse periods. Desert-adapted bulbs survive for years in a dormant state, not producing any aerial leaves until moisture is sufficient. Root succulents are less common, but they include plants like yams. Root succulents typically have non-succulent shoots and leaves that die back during drought, being replaced in the next moist season by new shoots that sprout from the root crown, a bit of shoot located at the top of the root. Wood succulents produce wood with a high percentage of parenchyma cells or a type of fiber that stores water. Examples are baobab trees, boojum trees, and elephant trees. Each of these alternatives has particular consequences. Water stored in wood is near vessels and may be especially effective in preventing cavitations, but it must then be transported out to leaves and flowers as in non-desert plants. Succulent leaves store water near photosynthetic tissues, keeping them hydrated and photosynthesizing even during very dry periods. Stem succulents may have thin, flat ordinary foliage leaves that are ephemeral, abscising when dry seasons start, after which the stems must perform all photosynthesis. Other stem succulents, such as cacti, never have large foliage leaves and must rely solely on their stems for photosynthesis. Such plants are always good at conserving water but can never take full advantage of occasional moist periods. A completely different way of tolerating drought is the capacity to survive protoplasmic desiccation. Many desert-adapted mosses have no capacity to store water, and they are perennial, not ephemeral. As the habitat becomes drier, so does their protoplasm until they become dormant for months. The water potential of their cells becomes extremely negative, which would kill most plants, but these are merely inactive, not dead. On days with sufficient dew, fog, or light rain, the mosses rehydrate in just an hour or so and quickly resume ordinary metabolism, including photosynthesis, growth, and development. Their activity will last as long as the environment remains sufficiently moist, but this might be just a few hours. Having a body water content that changes with habitat moisture is called poikilohydry. It also occurs in some liverworts, some species of Selaginella, the tiny cactus Blossfeldia, and a few other vascular plants. Lichens are arguably the supreme examples of poikilohydry, being capable of drying to very low water content for months without dying, even if growing on rocks exposed to full summer sun. Table 12, 6 Speed of xylem sap translocation. Figure 12.21 As water moves out of the leaf into the air, the tissues dry and a water potential gradient becomes established. Water flows from the xylem, where water potential is least negative, toward air, where water potential is most negative. Figure 12.22 If water can move laterally between two solutions, no lifting is involved. The slightest difference in water potential results in water movement. If water must move upward against gravity, work must be done. A slight difference in water potential may not be enough to cause water movement, only a large difference will. To overcome gravity and friction, the water potential of plant tissues receiving water must be at least 0.2 MPa more negative than that of roots for every 10 meters of height separating them. In the case illustrated here, with a difference of 0.15 MPa, water would not move up the stem. The water potential of the leaf would have to become 0.5 MPa more negative. Figure 12.23 This epiphytic cactus grows upside down. Its roots cling to the bark of a big branch of a rainforest tree, and its slender stems dangle straight down. Because its transpiration surfaces are lower than its roots, water flows downward from the roots, and gravity actually assists xylem conduction rather than hindering it, as in most plants. Returning to the plant in our example, transpiration causes leaf cells to lose water, and their water potentials become more negative. Water moves into them from tracheary elements, and tension pulls on water molecules in the xylem. When the water potential in the uppermost tracheary elements has become sufficiently more negative than that in the lower elements, friction and gravity are overcome and water moves upward. This causes the lowermost xylem cells to pull water inward from the root cortex, which in turn pulls water in from the root epidermis. Water potential of the root epidermis and root hairs becomes more negative than that of the soil, and water moves automatically into the root. Long-distance water transport occurs in this manner as long as the soil is sufficiently moist. A sandy soil that has 30% moisture has a water potential of approximately 0.001 MPa, almost equal to that of pure water. Water is held in soils by cohesion and adhesion as wedges and droplets between soil particles, and in sandy soils, root hairs can easily draw water from moist soils. Gravity and evaporation also pull water away from the droplets in sandy soils, so these soils dry quickly after a rain. As the soil dries, the most mobile molecules are removed, and those tightly bound to soil particles remain. In a dry soil, not only is less water present, but it is also relatively immobile. Clay soils are composed of thin flakes with a high surface-to-volume ratio. When wet, they hold a great deal of water, but it is firmly bound as a hydration layer. No root can pull water away from clay that is even slightly dry. Loam soils consist of sand, silt, and clay and have a diversity of pore sizes. During a rainfall, loam soils absorb large amounts of water and then hold it for weeks after the rain has stopped. The diversity of pore sizes allows root hairs to absorb water but prevents gravity from pulling the water so deep into the soil that roots cannot reach it. Table 12. 7 water potentials of soils. Many roots remain healthy with their water potentials as low as 2.0 MPa. They still absorb water from soils that are quite dry, but if the plant is 10 meters tall, the leaves would have to have a water potential at least slightly more negative than -2.2 MPa to overcome friction and gravity. This is also possible in some species, but typically, the leaves would either be dormant or preparing for abscission. When both soil and air are dry, plants are greatly stressed. Even if stomata close, transpiration continues, at least through the cuticle. Leaf water potentials become more negative, but water cannot move upward easily because the soil is so dry. Tension on the water columns increases, and at some point, cohesion is overcome, hydrogen bonding is broken over a large region, and the water column breaks. This breaking is called cavitation, and the water column acts just like a broken cable. Molecules above the cavitation point are drawn rapidly upward because they are now free of the weight of the water below them. Those below the cavitation point rush downward because nothing supports their weight. Between the two portions is space called an embolism, which expands until its surface encounters a solid barrier such as a pit membrane. The water embolism surface cannot pass through pit membranes, but it can pass through perforations because they are open holes. When an embolism forms in a tracheid, only that tracheid loses its water, but when an embolism forms in a vessel element, it may expand through perforation after perforation until the entire vessel has been emptied. Figure 12, 24 Many desert shrubs withstand severe desiccation. Even though their water potentials become extremely negative, the cells survive, although they may become inactive. This Nolina perryi is in full bloom despite its very dry habitat, Joshua Tree National Park. Plants such as this floating aquatic stream plant do not tolerate water stress at all, if their water potential falls very far below 0.2 MPa. They die quickly. During a drought lasting 8 months with no rain at all when this photograph was taken, the ground became so dry the bamboo died. However, the trees in the background were still healthy, and a few vining weeds climbing the dead bamboo were not merely surviving but actually growing. Figure 12.25 Severe tension can overcome the cohesion of water molecules and cause an embolism to form and expand rapidly. The embolism can pass through holes such as perforations but is stopped by pit membranes. If an embolism occurs in a tracheid, it cannot spread beyond the tracheid, but if a vessel element cavitates, the embolism spreads throughout the entire vessel. This is the trunk of a large water oak that was cut down. As the chainsaw cut through the xylem vessels, the water in them was cavitated, so now there is no hydrogen bonding to hold the water in the wood. The water here is pouring out of the wood, not just dripping, and the flow continued for almost a full minute. If this wood had been composed of tracheids, almost no water would have come out. Cavitation often means that tracheid or vessel can never conduct water again. The cohesive